Tissue culture is the in vitro aseptic culture of cells, tissues, organs or whole plant under controlled nutritional and environmental conditions, often to produce the clones of plant. The resultant clones are true to type of the selected genotype. There are various factors that determine the success of in vitro, such as explant sources, sugar sources, and plant growth regulators. As stated in the title, How to Produce Durian from Plant Tissue Culture. Based on researchers, the micropropagation of durian was done with employing various explants, such as apical and lateral buds, immature leaf, zygotic embryo, or knots. There is several techniques of plant tissue cultures. Pre-experimental phase. Firstly, we need to sterilize the room or culture room by fumigation or mopping with formalin. The glassware need to be washed using proper washing protocol. The media choice, for example, MS medium, white medium and gambox cell suspension. The medium must contain nutrient, carbohydrates, solidifying agent, water, growth regulator and special additive. And lastly, the preparation of stock solution with different composition of medium that prepare based on protocol and keep in refrigerator.
For experimental phase, it includes following steps. Media preparation. pH of the media should be 5.5 to 5.8. Next, sterilization of nutrient medium. Autoclaving the medium for 20 to 30 minutes. Collection of explants. A group of cells or single cell is taken, such as shoot tip, stem, leaf, root, and axillary bud. Next, it involves sterilization and inoculation of explants. The last one is incubation and observation. Incubation is carried out in incubation room. Proper light condition of 16 hours photo period, 8 hours dark period at temperature 25 plus 30 degrees Celsius and relative humidity of 50 to 60%. The last technique is post-experimental phase. It is about to transfer to greenhouse. Cultured plants are acclimatized or hardened in greenhouse after complete plant development. Healthy and tolerating plants are finally transferred to fields. Acclimatization or acclimation is the process of an individual organism adjusting to change in its environment, allowing it to survive changes in temperature, water, and food availability, other stresses, and often relates to seasonal weather changes. Acclimatization occurs in a short time, within days to weeks, and within one organism's lifetime.
For the method of plant tissue culture of durian plant, the first method is plant material, where the explant, which is the flower bud, is collected from the local durian trees. So the selection is based on the abundant number and the sterility of the explant were taken as a considerations of explant choice. The next step is surface sterilization. First, the flower bud will be rinsed with the tap water and wrapped up with hand and cotton bud. Then, they will be smeared with Clorox bleach until the scale is peeled slightly, and the remaining will be removed by cotton bud and left it for 30 to 50 minutes. Next, the flower bud will be washed with the tap water for 1 hour and put the flower bud into the laminar flow and dip it in the 96% of alcohol and flame it over Bunsen burner until the outer skin dries. Later, the dry skin was cut off and discarded, while the rest of the skin will be cultured in the media. Lastly, the observation all of these parameters will stand for every week in 6 weeks.
Assalamualaikum everyone, this is Ashraf and I'm here to explain to you about media preparation. The durian flower bud explants were germinated aseptically in basil media. The media we use is half-strength Russiagate and skook, which is known as MS. It is then solidified with 0.8% agar. The media is at pH 5.8. Then we add growth regulators, TD azuron and some sucrose. The media is then autoclaved for 20 minutes at 120 Celsius. Finally, pour 30 ml of media into each of 650 ml wide mouth jar where the plant tissue was cultured in the glass container with screw plastic lid. Now, on to culture condition. The durian explants were cultured and embedded into media. One bottle contained only one explant. It is incubated under cool white fluorescent light in incubation room, where it will receive 16 hours of light and 8 hours of dark with light supplied by cool white fluorescent lamps and cultured at around 25 degrees Celsius for 6 weeks. The illuminance is at around 1000 lux. Finally, we can conclude that in this experiment, the media needs to contain plant hormones necessary to stimulate development of differentiated tissues in order for the explants to grow. We also need to maintain as sterile an environment as possible so as not to contaminate the media with bacteria and spores of other plants. Since other propagation techniques such as budding, grafting, and cutting tend to damage the mother plant and potentially disrupt the production of the fruit, it is decided that propagation using in vitro technique is the most suitable one for durian plant. That is all. Thank you for listening.